Chapter 24 considers the chromosomal basis for inheritance. And according to the list of learning objectives that I received, these are the only two things we need to really take away from Chapter 24. But I'm going to give you just a little bit of background so that we understand what's going on with things like aneuploidy, non-disjunction, sex chromosomes, things like that. So I'm going to back it up a little bit and start by reminding us some of what happened in Chapter 23, how Mendel developed the law of independent assortment, saying that a pair of alleles are going to segregate independently from other alleles during gamete formation. So different genes, different characters can be separated from each other. So Mendel was able to separate the greenness and the yellowness character from the roundness and wrinkledness character. Now, he was able to do that because those genes, he didn't know about genes or chromosomes or any of that, but because those characters were on different chromosomes. So independent assortment only works in the same way that Mendel observed it when genes are on different non-homologous chromosomes, or if they're on the same chromosome, if they're at opposite ends of that physical chromosome. So genes that are physically located near to each other on the same chromosome tend to be inherited together because there is a physical connection between them. So remember I showed you this picture back in Chapter 23, where, again, this is not a P karyotype. This is a human karyotype. But just as a for example, the gene for that character, the roundness and wrinkledness, is on a separate chromosome from the yellow and the greenness gene that's on a separate chromosome. And that's how he was so able to fairly easily separate those two characters and see independent assortment. But genes may carry lots and lots of genes. Each one has to carry lots and lots of genes because there's only a limited number of chromosomes or places that you can put those thousands of genes that are required to make an organism. So what Mendel called hereditary factors, we would now call genes. And we know that genes are physically located on the chromosomes, and we can see where those genes are using some very sophisticated techniques. For example, we can put a label, a fluorescent label, onto a particular gene and see that here in this image, that bright yellow spot there and there, those are the same gene on two different sister chromatids, and the same gene is on the homologous chromosome. So one of these chromosomes came from mom, the other came from dad, and the same gene, the same instructions for a particular protein are encoded about the same spot near the centromere on those homologous chromosomes. That's pretty cool, don't you think? So Mendel didn't know a lot about what was happening at the molecular level. He wasn't looking for those kinds of things. He was looking at macroscopic, or things he could observe and count with the naked eye. And in fact, nobody really knew about mitosis and meiosis in Mendel's time. It wasn't until he was a very old man, or after his death, that mitosis and meiosis were identified, and the processes of cell division were able to be seen through the microscope. So the chromosome theory of inheritance that we now have as sort of the descendant of Mendel's ideas says that Mendelian genes have specific loci or physical locations within the chromosomes, and that chromosomes explain how we see segregation and independent assortment. So we're kind of combining Mendel with mitosis and meiosis. So for example, in the parental generation, in that dihybrid cross, when he crossed yellow round-seeded peas with green wrinkled-seeded peas, they produce their gametes and through fertilization. And what did they get? What did he see? You should remember this. He got all yellow round seeded plants because those were both the dominant characteristics. But in that F1 generation, they were all hybrids. They all had copies of the round and the green and the wrinkled and the smooth genes. So then during meiosis in the plants in that F1 generation, the two different alleles for each gene were separated during gamete formation. So some of the gametes were going to get round, some were going to get green, some were going to get wrinkled, some were going to get smooth. And the law of independent assortment says that if you have the genes on non-homologous chromosomes, so you see we've got a big set and a little set, if those genes are on different chromosomes, they're not going to necessarily travel together. So we might get a round gene and a green gene going to one gamete, and a yellow gene and a wrinkled gene going to another gamete. And that explains how he got the ratios that he observed in the F2 generation of 9 to 3 to 3 to 1. So 9 being double dominant, 3 being one dominant character and one recessive, 3 being the other character recessive and dominant, and then 1/16th of all of these P's being double recessive. All right, so along comes another researcher in the early 20th century named Thomas Hunt Morgan. He worked with a different model organism. Instead of peas, he used the much less delicious fruit flies. But while fruit flies are not a very valuable commodity, they're not something that we want to have in our kitchen or anywhere around us, really. They did serve as a good genetic model, a model organism. They're real easy to raise. I mean, they're hard to get rid of. So it makes sense that it'd be easy if you were trying. You could raise millions and millions of these little guys. So the work that Thomas Hunt Morgan did was with this very famous fruit fly, which has the scientific name Drosophila melanogaster, or the dew-loving black-bellied fly, is what that name means. So why were they a good model organism? Well, they reproduce like crazy. A single pair of parents could produce hundreds of little baby flies in a very short amount of time. It only takes 2 weeks for them to go from being eggs to being adult flies. So you get lots of data in a very short amount of time. They only had 4 pairs of chromosomes, and that's important for reasons that we're not going to get into. But as far as finding linkage, the fewer chromosomes you have, the more likely that genes are going to be linked together. And the logic goes something like this. If you have a whole household full of stuff to pack up, and you only have 4 boxes, it means the likelihood that 2 of the things that you need are going to be in the same box is a lot higher than if you had everything packed up into, say, 46 boxes, and you were trying to find your toothbrush and your underwear and your rolling pin. The likelihood that they're all going to be together in the same box is a lot more likely if you only have 4 boxes versus if you have a lot of boxes. And like people, fruit flies have this sex determination schema of X and Y. So female fruit flies are XX, just like human females, and male fruit flies are XY, just like in human males. That's not universal throughout the animal kingdom, but fruit flies do have this method of determining sex, and that was helpful too. That was really the most important thing from Thomas Hunt Morgan's early work, showing that genes were physically located on chromosomes. Because we knew that the arrangement of chromosomes, those X and Y chromosomes, determined whether a fly was going to be male or female. So what Thomas Hunt Morgan did was he took the fruit flies and he exposed them to mutagens, or agents of mutation. And in this sense, mutation just means he's changing the DNA, changing the genes of these fruit flies, and kind of seeing who survived and what kind of mutations he could produce. And one of the first mutations he produced was a fly that had white eyes instead of red eyes. And the fact that the mutant mosquitoes had white eyes suggested that something had happened to their DNA. So he tried to test Mendel's law of independent assortment. So what did he do? He mated male flies with white eyes with female flies that had red eyes. And in the naturally occurring flies, he called those the wild type. So if it had a characteristic that was just like the ones that he collected from nature, that's what he called wild type. And the white-eyed flies were mutants that he had generated by zapping them with DNA-harming substances. And again, these are fruit flies, so nobody from PETA is going to, was around to say, hey, you shouldn't be mean to those fruit flies. But ethics aside, what did he find? He found that in the F1 generation, that they were all red-eyed, suggesting that that white-eye mutation was recessive to the red-eye trait. So then he did the F2 cross between the flies, the red-eyed flies in the F1 generation, and just like Mendel would have predicted, he got a 3-to-1 ratio of red-eyed to white-eyed flies. But, and here's the kicker, only the males had white eyes. None of the females had white eyes. So, nobody really cared that much about eye color in flies, but the significant discovery here was that since that red-eye trait only appeared in males, it followed that it had to have something to do with the sex chromosomes. And it stood to reason that that white-eyed mutation must be on the X chromosome, and therefore genes that encode things like eye color must be physically located on the chromosomes. Okay, so how does this work? Here's sort of a cartoon documenting that. So in the parent generation, red-eyed and white-eyed flies. In the F1 generation, males, females, everybody had red eyes because it's the dominant character. He let flies of this generation breed with each other, and what did he observe? A 3-to-1 ratio, but half the males had white eyes, half the males had red eyes. All of the females had red eyes. So what did he conclude? So here it's a little bit different from the uppercase and lowercase way of drawing things that we saw in the last chapter. And I'm sorry, but Thomas Hunt Morgan was not using Mendel's way of drawing things. So this little W plus means that's the wild type gene, and this little W with no plus means that's the mutant gene for white eyes. So what he reasoned was that the males could pass on either an X chromosome or a Y chromosome, just like our dads can pass on either an X chromosome or a Y chromosome. Our mothers, who have 2 X chromosomes, can only pass on an X chromosome, which meant that at the end of the experiment, only the males who only had 1 copy of that white allele would have the trait. And females, because it's a recessive characteristic, they would get 1 copy of the white allele from dad, but it would be masked by the red allele from mom. So very important discovery by Thomas Hunt Morgan that genes actually are physically located on chromosomes. They're a physical thing. We know now that they're DNA sequences of nucleotides, but that's kind of getting ahead of ourselves. So, experiment, the results, and the conclusions.